Penicillin-binding proteins. Penicillin-binding proteins (PBP) are a group of proteins that are characterized by their affinity for and binding of penicillin. They are a normal constituent of many bacteria. The name just reflects the way by which the protein was discovered. All B-lactam antibiotics except for tabtoxinine-β-lactam, which inhibits glutamine synthetase, bind to PBP, which are essential for bacterial cell wall synthesis. PBP are members of a subgroup of enzymes called transpeptidases. Specifically, PBP are DD-transpeptidases. Diversity. There are a large number of PBP, usually several in each organism, and they are found as both membrane-bound and cytoplasmic proteins. For example, Spratt 1977 reports that 6 different PBP are routinely detected in all strains of E. coli ranging in molecular weight from 40,000 to 91,000. The different PBP occur in different numbers per cell and have varied affinities for penicillin. The PBP are usually broadly classified into high molecular weight HMW and low molecular weight LMW categories. Proteins that have evolved from PBP occur in many higher organisms and include the mammalian lac protein. Function. PBP are all involved in the final stages of the synthesis of peptidoglycan, which is the major component of bacterial cell walls. Bacterial cell wall synthesis is essential to growth, cell division, thus reproduction, and maintaining the cellular structure in bacteria. Inhibition of PBP leads to defects in cell wall structure and irregularities in cell shape, for example filamentation, pseudomulticellular forms, lesions leading to spheroplast formation, and eventual cell death and lyses. PBP have been shown to catalyze a number of reactions involved in the process of synthesizing cross-linked peptidoglycan from lipid intermediates and mediating the removal of D-alanine from the precursor of peptidoglycan. Purified enzymes have been shown to catalyze the following reactions: D-alanine carboxypeptidase, peptidoglycan transpeptidase, and peptidoglycan endopeptidase. In all bacteria that have been studied, enzymes have been shown to catalyze more than one of the above reactions. The enzyme has a penicillin-insensitive transglycosylase N-terminal domain involved in formation of linear glycan strands, and a penicillin-sensitive transpeptidase C-terminal domain involved in cross-linking of the peptide subunits, and the serine at the active site is conserved in all members of the PBP family. Some low molecular weight PBP associate with the mREB cytoskeleton and follow its rotation around the cell, inserting peptidoglycan in an oriented manner during cell growth. In contrast, high molecular weight PBP are independent from mREB and maintain cell wall integrity by detecting and repairing defects in the peptidoglycan. Antibiotics. PBP bind to B-lactam antibiotics because they are similar in chemical structure to the modular pieces that form the peptidoglycan. When they bind to penicillin, the B-lactam amide bond is ruptured to form a covalent bond with the catalytic serine residue at the PBP active site. This is an irreversible reaction and inactivates the enzyme. There has been a great deal of research into PBP because of their role in antibiotics and resistance. Bacterial cell wall synthesis and the role of PBP in its synthesis is a very good target for drugs of selective toxicity because the metabolic pathways and enzymes are unique to bacteria. Resistance to antibiotics has come about through overproduction of PBP and formation of PBP that have low affinity for penicillins, among other mechanisms such as lactamase production. These experiments changed the structure of PBP by adding different amino acids into the protein, allowing for new discovery of how the drug interacts with the protein. Research on PBP has led to the discovery of new semi-synthetic B-lactams, wherein altering the side chains on the original penicillin molecule has increased the affinity of PBP for penicillin, and thus increased effectiveness in bacteria with developing resistance. Presence of the protein penicillin-binding protein 2, PBP2, is responsible for the antibiotic resistance seen in methicillin-resistant Staphylococcus aureus MRSA. The B-lactam ring is a structure common to all B-lactam antibiotics. Other images.